Richa Purak. Today I like to share some information about messenger RNA. Messenger RNA is a single stranded RNA. The messenger RNA carries genetic information from DNA to cytosol where it is used as a template for protein synthesis. Messenger RNA is furnished with genetic code, a sequence of codons. Genetic code is universal, comma-less, non-overlapping, degenerate, and non-ambiguous. The message of RNA is read consecutively in 5' to 3' direction. Each amino acid is recognized by a specific codon. Messenger RNA comprises about 5% of the total RNA in the cell but is most heterogeneous type of RNA in cell varying in coding region. Only a small amount of DNA is used in coding for coding of messenger RNA. This slide shows messenger RNA furnished with genetic code. The genetic code is the total of codons, and each codon is a triplet which is recognized by a transfer RNA by virtue of its anticodon, and it also recognizes the amino acid. Messenger RNA, as other RNAs, is synthesized by transcription using DNA template. In prokaryotes, there is only one RNA polymerase enzyme for transcription of all the three RNAs. But in eukaryotes, there are three different RNA polymerase enzymes as RP1, 2, and 3. RP2 transcribes messenger RNA. The DNA double helix opens locally by RNA polymerase and transcription factors. During transcription, only one strand is used as template, antisense strand. The RNA produced has base sequence similar to other strand known as sense strand. This slide shows how transcription takes place using one of the strand of DNA double helix, and the RNA formed has base sequence like the sense strand. This slide again shows the in eukaryotes transcription takes place in the nucleus, and then after transcription messenger RNA moves to the cytoplasm where it take part in protein synthesis with the help of ribosome and transfer RNAs. Messenger RNA is short-lived, between several seconds to few minutes in bacteria, but longer-lived in eukaryotes, possibly because it has to move out of the nucleus to the site of protein synthesis in the cytoplasm. In prokaryotic organisms, ribosome can attach to messenger RNA while it is being transcribed. In this situation, translation begins at the 5 end of messenger RNA, while the 3 end is still attached to DNA. Prokaryotic messenger RNA at its 5 end has a signed-Delgarimo sequence which is rich in purine nucleotides and help binding of messenger RNA to the smaller subunit of ribosome by means of its single ribosomal RNA. Prokaryotic messenger RNA undergoes very little processing after transcription. In prokaryotes, transcription and translation are coupled, but in eukaryotes, a little processing occurs after transcription. A cap of ATP or GTP is added at the 5 end after transcription. Guanine or adenine get methylated for it. Cap facilitates binding of messenger RNA with ribosome and provides stability. Cap is followed by non-coding or UTR region 1, which is used for binding of messenger RNA with smaller subunit through its single ribosomal RNA. Initiator codon AUG follows UTR 1, which directs to introduce methionine at the first position in polypeptide chain. It is followed by coding region 1200 to 1500 nucleotides and can code for polymerization of more than 500 amino acids. Towards the 3 and one of the 3 termination codons provides signal for termination of polypeptide chain. On the 3 end, a polyadenine tail is added. after transcription, which provides stability to messenger RNA. Precursor eukaryotic messenger RNA possess both coding and non-coding regions and require some processing to give mature functional form. Now this slide shows the upper diagram is a prokaryotic messenger RNA having signed Delgarimo sequence at the 5 end helps in correct orientation of messenger RNA on the smaller subunit of ribosome by making hydrogen bonds between purine and pyrimidine nucleotides. This is eukaryotic ribosome, and this is also eukaryotic ribosome showing cap on the 5 end, tail on the 3 end, and non-coding region followed by initiator codon. Then there is the coding region, then there is the termination codon. Now in eukaryote, processing of messenger RNA takes place, and the processing can be of three types: 5' capping, addition of poly A tail, removal of introns. The cap is a 7-methyl guanosine attached backward to the 5' terminal end of messenger RNA, forming unusual 5' 5' triphosphate linkage in place of 3' to 5', catalyzed by nuclear enzyme guanyl transferase. Methylation of this terminal guanine occurs in the cytosol by guanine 7-methyl transferase. Source of methyl group is S-adenosylmethionine. The cap permits initiation of translation and also provides stability of messenger RNA. This slide shows the cap and the tail. Cap has 3 phosphates. Addition of a poly A tail. Most eukaryotic messenger RNA have a chain of 40 to 200 adenine nucleotides attached to the 3 end. This poly A tail is not transcribed from DNA but is added after transcription by the enzyme polyadenylate polymerase. Poly A tail also provides stability and facilitates the exit from the nucleus. After messenger RNA enters the cytosol, the poly A tail is gradually shortened. Removal of introns. Maturation of eukaryotic messenger RNA usually involves the removal of RNA sequences which do not code for protein, intron, or intervening sequence from the primary transcript. The remaining coding sequence exons are spliced together to form mature messenger RNA with continuous coding stretch. The removal of introns and splicing of exon is helped by small nuclear RNAs in association with proteins. This slide shows how the introns shown in blue color are removed and the exons shown in red color are spliced with each other to form a continuous coding stretch. Now, generally messenger RNA is linear, but occasionally the 5 end and 3 end of messenger RNA may interact, that is cap and tail, and become circular. Circularization promotes recycling of ribosome on same messenger RNA, leading to efficient translation. They are not dissociated. Degradation and half-life of messenger RNA. We know that messenger RNA is short-lived. Messenger RNA is transcribed as a complementary copy of DNA that eventually translates a polypeptide chain. Messenger RNA travels from nucleus to cytoplasm for translation. Because messenger RNA is constantly being degraded in the cytoplasm, it is synthesized at a much higher rate than necessary for maintenance of steady amount. Different messenger RNA cells have different lifetime. In bacterial cells, individual messenger RNA can survive from few seconds to more than an hour. With longer survival are able to produce more polypeptide chains of the same time. Short half-life of messenger RNA enables cell to produce altered protein in response to changing needs. There are different mechanisms for degradation of messenger RNA in prokaryotes and eukaryotes. Eukaryotic messenger RNA acquire a longer stability due to addition of 5 cap and 3 poly A tail. In some instances, many ribosomes can bind to the same messenger RNA successively, all producing similar polypeptide chain or differing in length. The structure is known as polysome or polyribosome. Messenger RNA is used repeatedly to meet high demand of specific protein in a cell. The messenger RNA can be monocistronic or polycistronic. If it codes for only one polypeptide chain, it is monocistronic, as in eukaryotes. In prokaryotes, generally they are polycistronic, coding for more than one polypeptide chain, but are related and are transcribed by structural genes clustered in a group known as operon and are regulated as a unit. Some messenger RNAs have signal codons just after initiator codon on the 5 end, and because of this signal codon, signal sequence appears in the amino terminus of polypeptide chain, growing polypeptide chain, which is recognized by SRP and directs the ribosome to bind with endoplasmic reticulum surface, and protein synthesis continues in its lumen, resulting in secretory protein. Those messenger RNA which do not contain the signal codon completes protein synthesis after binding to the ribosome free in the cytoplasm, give rise to structural protein. This slide shows how signal sequence directs the protein targeting. Then this again shows signal sequence directing protein targeting, and this is the two type of protein synthesis, one on ribosomes free in cytosol and other ribosomes attached to endoplasmic reticulum. With this, the class is completed. Thank you.